A quick recap of mitosis. Mitosis is the process, it's a part of cell division where a cell takes its copied DNA and divides that into two new nuclei to get distributed into two new cells. The new cells are called daughter cells. And so in this picture here, you can kind of see they're not really showing the chromatin. They're showing maybe some parts of chromatin, which is just a different form of the DNA when it starts to condense and coil. When normally you don't see DNA, it's normally uncoiled and just kind of loose within the nucleus. But when it gets ready to divide, the DNA starts to coil. So when it actually gets coiled, you can see the little tight coils forming here, and it will actually form these structures called chromosomes. And if a cell is getting ready to divide, at the end of interphase, the chromosomes have already doubled. So the centromere attaches the chromosomes. So here are the chromatids. So here's one, here's the identical one, here's another short end, and here's the identical one here. So 1, 2, okay? And these are within the nucleus. So let's officially go over the stages of mitosis and the whole cell cycle. So in interphase, a cell grows, a cell carries out its normal functions. If it's a heart cell, it's pumping blood. If it's a nerve cell, it's sending signals. But at the end of its life cycle, or the growing phase, it's going to prepare to divide. In order to do that, the centrioles copy themselves and the chromosomes start to condense. They've already been copied when they start to do that. So an entire copy of the genetic material has happened, and now we enter the first phase of mitosis called prophase. Again, what's going to happen in mitosis is this jumbled copied DNA is going to get separated into two new nuclei. So in prophase, we see that the chromatin has fully condensed into chromosomes, and the chromosomes are actually connected sister chromatids because we have the copies connected to one another. So each of these represents a copied set of chromosomes. The centrioles are moving to opposite ends. They're producing spindle fibers. And the last thing that's not as clear is this nuclear envelope is breaking down. Because if we're going to move all these chromosomes all over the place and separate them, we can't do that in this confined space. This needs to break apart. Now we enter the second phase of mitosis, which is called metaphase. And the distinguishing characteristic in metaphase is that the sister chromatids are moved down to the middle, called the metaphase plate. Notice the spindle fibers are attached to these centromere points, and they'll be used to pull each copy of the chromosomes to opposite ends of the cell. When that starts to happen, we've entered the next phase called anaphase. The sister chromatids have been pulled apart, and they're moving to opposite cells. To make that happen, the spindle fibers start to shorten. Also worth noting is that the cell is starting to pinch, right? This is like a whole process. It happens, in a fluid motion. So it's kind of hard to take a snapshot and say, that's it, snapshot and say, that's it. It does progress from here to here, kind of like a freeze-framed little pictures at a time. Okay, the distinguishing feature of the next phase, telophase, is that we formed the two new nuclei. So we go from this to this. And for that to happen, the spindle fibers have to disappear, because once we've moved the sister chromatids, we don't need to move them anymore, so they would disappear. The nuclear membrane reforms, and now we have two new nuclei, each with a set of chromosomes. Mitosis is complete. Cell division is not complete. We see it's going towards completion, and that the cells have pinched off further, more so than they did in anaphase. But we need another phase to finish cell division. Mitosis is done in telophase, but the last phase of cell division is cytokinesis, where the cytoplasm gets divided along with the other organelles like ribosomes, endoplasmic reticulum, Golgi apparatus, and whatnot. And the cell membrane has completely pinched off, forming those two new cells. So we started with one cell. We now have two new complete cells, each with an original set of chromosomes. Worth noting here is that if the cell was carrying out its normal cycle, let's say it had 10 chromosomes, by the time it got to the end of interphase, it would have 20, two copies of 10. Those two copies of 10 would get divided into two new cells. So now these two nuclei each have their own set of 10 chromosomes. And eventually we get two new cells that each have 10 chromosomes, just like the original cell we're not seeing here. And that's the end of mitosis. Just to talk about a few more things, I don't really care about the question here, but I want you to see that mitosis is a very short part of the cell cycle. Cytokinesis even shorter. All of this together would be cell division. The preparation of cell division would be the copying of the chromosomes here, and the centrioles copying the chromosomes, condensing here. And this is growth and involves all of the other cell activities that have to happen, right? So a very short time really for cell division. Okay, and I just want to fast forward and just talk about DNA a little bit. So DNA is the molecule, the nucleic acids that make up the chromatin when it's just condensing. The condensed chromosomes, which are those X-type structures that we saw in the last couple of slides. And we'll get more into talking about these nitrogen bases and how they pair together later on. I just wanted you to see that the DNA does replicate in this way. And when we replicate the DNA, we get two copies. And if all of this DNA is what makes up those chromosomes. Real quick, just to recap the different stages of the cell cycle. This is interphase. The cell's growing, making a copy of its DNA at the end of interphase. And now we're entering mitosis. So this is prophase. And chromosomes and spindle fibers form. Nuclear envelope breaks down. Now we're in metaphase. Chromosomes line up across the center. And the chromosomes are attached to the spindle fibers at the centromere. Now we're going to anaphase. The centromere is split. The chromatids separate, move to opposite ends. And now we're at the end of mitosis in telophase. Chromosomes stretch out. That means they unwind. They form loose chromatin again. And the nuclear membrane reforms. Mitosis is finished. Cell division is not. We still need cytokinesis, where the cell pinches in two, and each daughter cell has the same number of identical chromosomes as the original cell that went through its life before it copied its DNA. One more thing to talk about, and that's cancer. Why do we talk about cancer when talking about mitosis? Because cancer is defined as uncontrolled cell growth. So you have all these cells that are skin cells or any kind of organ cells, and all of a sudden you get a cell that starts growing out of control. That cell is cancerous. What happens with these uncontrolled cells is they then form masses or tumors. So why is cancer a problem? Doesn't matter what organ this is. Let's just say it's the lung, and the function is to help you breathe, right? Expand to take in oxygen and expel carbon dioxide. Well, these cells here are doing that job. These cells do not. They are still your cells. They just don't function anymore like the organ they're supposed to. So they're growing uncontrollably. They're just not able to do the job that they were intended to do. So what happens if this mass gets big enough in your lungs, you can no longer breathe. And that shuts down your body and you can perish from that, right? The other really important thing to understand with cancer is that these cells that grow uncontrollably spread. So they can break off, they can get into the bloodstream, and they can travel to other organs, creating other masses throughout the body. So, again, this is related to mitosis and cell division because cancer is defined as uncontrolled cell growth. And the reason cancer is so hard to treat is because these are your cells. So any treatment, whether it be chemotherapy, radiation, you are trying to target the cancer cells, which are your cells that you don't want, but it's hard to differentiate them from the other cells around them that you do want. And so oftentimes, the chemotherapy and other treatments will affect your body, will affect your healthy cells. So really the best treatments we are starting to move away from this to more targeted therapies, is how do we affect the cancer cells without affecting the normal cells. Okay? And that's it.